J. Okay. So many windows open. J. All right, I can hear my echoey voice. That means we're good. All right. Okay. So we left off talking about heredity. We've gone through the first couple slides. So we had defined some terms. So basically we had defined our ideas of genotype and phenotype. Genotype being the genes and the alleles that are encoded on a homologous pair of chromosomes. Phenotype being the actual trait or characteristic that is expressed. We'd also talked about this idea of dominant versus recessive alleles, where a dominant allele, whenever it is present, is expressed. So in this case, this capital S, colored in orange, is going to our purple flower color. So we can see that whenever that's present, it's expressed. A recessive allele is only visible when there are two copies of that recessive allele. Recessive alleles are lowercase, so that's our white flower here. And we talked about the fact that we can refer to genotypes as homozygous, heterozygous, and homozygous again. So we have a homozygous dominant. We have a heterozygous, hetero meaning different. So we have one of the dominant allele, one of the recessive allele. We have a homozygous recessive, meaning two copies of this recessive allele. This slide is just telling us that again, and we talked about this and we went through an example problem as well. Okay, so we noted that if we have a recessive allele, the only time we'll see it, in this case our flamingo pink hair, the only time we'll see it is if we have two copies of that flamingo pink hair gene. So let's pick up, I don't think we did this one yet. If we did, then it'll be easier than usual. So let's try a second example. All right. Okay, looking good. Okay, so we're looking at this trait mohawk hair. It's a dominant phenotype, which means it's encoded by a dominant allele. So if that allele is present, it will be expressed. So if we're thinking about what genotypes have mohawk hair, anytime we see a capital H, which we see in both of these, right? So both the heterozygous individual and the homozygous dominant individual would express this dominant trait for mohawk hair. We can mostly skip over the history of these findings, although I know your book goes into them in some detail. But it is the case that some diseases are more complex. And often when we're thinking about diseases, we want to be careful that we know what chromosome they are transmitted on. And that is because the inheritance of our normal chromosomes, our autosomes, are the same for males and females, but specifically traits that are carried on sex chromosomes, so the X and the Y chromosome, are going to be inherited differently in males and females. So autosomes in a human are these chromosomes here. We see our chromosomes 1 to 22. Those are autosomes. They have numbers. Our sex chromosomes are the X and the Y in humans. Okay. So your average female is XX, your average male is XY. There are lots of different things physiologically that can do alterations to that, but that's the typical. Okay, so autosomal chromosomes, you have two copies. They're identical in terms of genes. So like if you have an autosome and it carries hair color, you might have different alleles on this autosome, right? You might have red on one and brown on the other, but they both carry a hair color gene. This is different than the sex chromosomes. You'll notice that with the sex chromosomes, the X chromosome is much longer than the Y chromosome. So this is why we differentiate between autosomal inheritance and sex-linked inheritance, is because you do have genes on the X chromosome that are not on the Y chromosome. So in males, some traits, specifically the ones carried on sex chromosomes, will only have one copy of that gene. But in autosomal inheritance, you have two copies of the gene, potentially two different alleles, and it works the way we were looking at with those funny little hair traits with our P's that we were looking at. So when we do Punnett squares, our normal Punnett squares, we're talking about autosomal chromosomes. So autosomal chromosomes, you have two copies on the homologous pair. Each individual carries two copies of the gene. Individuals with two identical alleles are homozygous. Individuals with two different alleles are heterozygous. So this lines up just with these particles inheritance, showing us really how it is on the chromosomes. Okay. We should note that plenty of traits or diseases in humans do work the way that we trace out with Punnett squares and just this sort of simple, what we call Mendelian principles. But there are lots and lots of traits and probably most diseases that are multigenic or have more complex genetics, where you have several genes on several different chromosomes that kind of add up to a full picture. So what we're working through for the purposes of our course are just the simplest examples. But in the future, if you learn more about genetic diseases, you'll see that it gets a lot more complicated than this. Okay. So we're going to work through an example that is pretty simple. So cystic fibrosis is a disease of, largely affects the lungs, but in general, kind of epithelial tissue. It's a recessive disorder. So let's say our allele combinations for cystic fibrosis are capital F, lower f, and lowercase f, lowercase f. So who would be affected in this situation with cystic fibrosis? Okay, looking good. Yeah, so when we talk about recessive disorders, I'm going to end the polling real quick. Okay. So when we talk about a recessive disorder, and in this case, we're talking about a recessive autosomal disorder, the only individuals that you'll see affected by the actual disease, if it's a recessive autosomal disorder, are the homozygous recessive individuals. So that's answer C. It is the case, however, that we will often be interested in people who are heterozygotes, right? Not because they express the disease themselves, but these are the individuals we're going to think of as carriers for a trait. So they don't have the disease, but because they have one copy of this recessive allele, their children may have the disease, depending on what the other parent looks like. Right. So when we're thinking about recessive diseases, people who have it, who are affected, are homozygous recessive, but we will have heterozygotes who are carriers for a disease, despite the fact that they don't have the disease themselves. Sorry about my dog, everybody. Go to bed. Must mean my mail's coming. So let's differentiate this from a dominant autosomal disorder. So familial hypercholesterolemia, which is basically a genetic high cholesterol, is a dominant disorder. So it's a dominant autosomal disorder. So now let's think about who would be affected if we have a dominant autosomal disorder. And here we put it in terms of some of our vocabulary words, right? Instead of just writing out the traits. Okay. So because we're now looking at a dominant disorder, so we have a dominant disorder, this means that any individual who contains a copy of this dominant gene, this dominant allele, are going to be affected. So this means both our heterozygous individual, right? Heterozygous individual is the one that has two different versions, but they do have one version of this dominant allele. They're going to be affected. But so is the person who has two copies of our dominant allele. So in this case, you would not expect to see any carriers for the gene who are not affected. There's no one who secretly has this gene, because when it's present, it is expressed. So that is a difference in the way we talk about recessive versus dominant disorders, is we know when we have this dominant allele present, even in the heterozygotes. Okay. So Punnett squares are our easiest way to determine our genotype and phenotype ratios from crosses, which is, from predicted offspring from sexual reproduction. So recommend that anytime you aren't sure what you're going to expect in the children, you sketch up a Punnett square. Okay, so here we have an example of how you'd set it up. All right, so if we have a Punnett square, let's imagine we have this gene, this G gene. We want to see what would happen if we have a homozygous dominant individual have children or have offspring with a heterozygous individual. So what we do first is that we're going to split up these two alleles. And this is really cool because as we saw in meiosis, right, this is actually physically what happens during meiosis when we make gametes, right? We made copies of these homologous chromosomes. We separated the homologous pairs, separating, right, this chromosome, this half of the pair from this half, right? So we are literally doing that as we do these rows, right? So we're going to put G for this heterozygous parent. I'll put big G over here for our homozygous parent. So then as we do the Punnett square, we type in, right, big G. So we're just putting what's on the row and what's in the column. So here, again, we have a big G, we have a little g. Here we have a big G again. And here we have a big G and a little g. Okay. So in this cross, where we have a homozygous dominant individual who is reproducing with a heterozygous individual, we can then express the probability that their children will have any given combination of alleles by looking at this square. So here we can see that, right, 2 of these 4 individuals are homozygous dominant, and we can reduce that expression, right, to be 1/2. So 1/2 or 50% are GG. And then we look at our other possible option. We got this other result, heterozygous, right? So again, 2 out of 4 which is the same as 1/2, are heterozygous. So a thing we will often want to do is to be able to create this Punnett square for a cross, but we also want to be able to recognize some typical ratios. Specifically, when we want to recognize ratios, if we're trying to do this quickly, we want to think about crosses with respect to the heterozygous individual, because sometimes we'll only be able to observe a phenotype. So a heterozygous individual will express a dominant phenotype, but secretly is carrying this recessive allele, right? So sometimes we will want to test that. We will want to know that. So we'll do a test cross, right? So sometimes you will be given like the distribution at the end. And when I say you will be given, I mean like in the literature, or if you were looking at family history or something. And you will want to be able to predict people's genetics from the resulting offspring. So for that one where we crossed a homozygous dominant individual with a heterozygous individual, we saw that we ended up with a 50-50 ratio of genotypes. If we did this with a homozygous recessive individual, we would see a 50-50 distribution of phenotypes, right? Because 50% of the offspring would end up with the recessive, homozygous recessive trait. We would be able to tell that apart from what happens if you cross, for example, two heterozygotes. When we cross two heterozygotes, we get this 1 to 2 to 1 ratio of homozygous dominant to heterozygous to homozygous recessive. And if we look at the phenotypes then, 75% highlighted in blue are the percent of individuals who show this dominant phenotype. 25 is the percentage who show this recessive phenotype. Okay. So we'll do some practice with crosses, making sure that we get familiar with some of the very typical combinations. But this heterozygous by heterozygous might be a good one to just kind of memorize if you feel like memorizing anything. So what we're going to do now, this is not part of our problem set, but we're going to go ahead and do some example problems to break this lecture up. So I'll put this in the chat as well. I'm going to stop sharing my screen for a second. So we're not going to do all the problems for each group, but you're going to work on one problem and then we'll come quick and I'll run through all of them together just so we get some practice here. I'll grab that link and put it in the chat. So here's the link. Do a problem with your group in your breakout room. I had some groups listed on the slide. It doesn't really matter, and you don't have to worry about pasting in the Punnett squares. We'll just go over them as a class. So just pick one and practice. And if you get to more, you get to more. I am muted. All right, so hopefully doing just one of those was pretty simple. Let's go through them. So in this first problem, we see we have a carrier individual, which we know means a heterozygote, and we have a wild type individual, which is here defined for us in this first problem as a homozygous dominant individual. Okay, so what I've done is I made us little Punnett squares for each of these. And for each of them, I'm splitting up our gametes for mom on top and dad down the side. Okay, so in this case, we distribute our carrier, so a dominant and a recessive allele, wild type, dominant and a dominant allele, and we go through. So we had capital F, lowercase f, capital F, capital F, lowercase f, for a risk of having our recessive disorder, right? Risk of cystic fibrosis is recessive. Because of this homozygous dominant individual, our risk of actually having this disorder is 0%. None of these are lowercase f, lowercase f. So we can compare this to if we have both parents as carriers, right? So here we split them up, dominant allele, recessive allele for one parent, dominant allele, recessive allele for the other parent. We're going to get capital F, lowercase f, capital F, lowercase f, and lowercase f, right? We're just distributing rows and columns. All right, so we see this 1 to 2 to 1 ratio for genotypes. And we can see that one of them here has cystic fibrosis, has this dual copy of the recessive gene. So our risk here for cystic fibrosis is 1 out of 4, which is 25%. So if we have our mom who has CF, right, has it, right? CF is the name of the disorder. We know if they have it because it's a recessive disorder, this means that we have 2 copies of this recessive allele. Dad is wild type, which is defined in that first problem as homozygous dominant. All right. We know every individual here is going to be heterozygous because they're always going to get a copy of the dominant trait from dad. They're always going to get a copy of this recessive trait from mom. So again, they're all carriers for this recessive gene, but none of them actually have the disease. Meanwhile, if we have a parent who has CF, having CF means 2 copies of the recessive. Dad is a carrier is a heterozygote. All right, so we got capital F, lowercase f, capital F, lowercase f, lowercase f, lowercase f, lowercase f, lowercase f. See that if a parent with CF has children with a carrier for CF, half of those kids may end up with CF. So we have a 50% risk of a child with CF because we have 2 out of 4. Finally, if mom and dad both have CF, there's only that recessive allele to be given. So every one of those offspring is going to end up with CF. So that was kind of rapid fire, but having done one, hopefully you can see how you would do the others as well. So that's really all we're doing when we do these Punnett squares. We're not going to worry about exactly how to read pedigrees for this semester, but just know that when they ask you at the doctor's office about family histories for like certain genetic diseases, what they are trying to do is build basically a chart of your family showing who has that disease and who doesn't. And using this chart, you can try and figure out someone's genotype the way that we just did in those examples, but backwards, and then help you evaluate your risk. Now, they don't always have to actually do the physical drawing, right? Your doctor probably isn't sitting there making the drawing. They have software that calculates the probabilities for them. But this is the basics of how it would work. So if you ever see one, we have circles for females, squares for males. And if we see the trait, we darken the square or the circle. So we're not going to worry about this whole pedigree, but it is a way you can analyze how these traits are passed down in families. Okay. Now, again, we're not going to worry about doing these for the purposes of this semester because we don't have a ton of time. But I do want you to be aware that not all inheritance is super simple. Specifically, stuff on sex chromosomes gets weird because we have these males who don't have 2 copies of the gene. So we will have different inheritance patterns in men for sex-linked or X-linked traits. And there's a lot of details in the slides about what that would look like, but don't worry about those for this semester. Right? But you could draw them out, and we can see how individuals who are XY versus individuals who are XX have a different proportion of alleles here. Okay. Other more complex inheritance patterns involve some traits that are coded for by multiple alleles, right? Like with your blood type, there aren't just 2 options. And we have type A blood, type B blood, and type O blood. And each of those have several genotypes that can code for them, basically. Well, not each of them, but some of them. So basically, sometimes we're going to have more options than this. And anytime we have more options, it's going to make it more complicated. In situations where we have multiple alleles like this, sometimes we're going to have alleles that have an order of dominance. Sometimes we're going to have alleles that are co-dominant. And this is basically what it looks like for blood type. So anytime we have a gene for protein A or a gene for protein B, both of them are going to be expressed. So like A isn't dominant to B, B isn't dominant to A, which is why some people have AB blood type, but both of them are dominant to O, because O is really a 0. It's for no protein expressed. All right, so we can have more complex ways that genes interact with each other is the takeaway here. Okay. So those of you who completed and are satisfied with your heredity problem set, you're free to go, but the rest of us are going to open up those heredity problem sets and work on a couple more examples.